Good morning, everyone. And it's a Sunday morning, right over. I'm from Chennai, India, and I welcome all of you to today's webinar, Saliva Annex. Well, not wasting any more time, I will just share my screen with you and let's plunge into saliva. Okay, so let me just give you an introduction of myself. I am Dr. Gargi Roy Goswami, and I am an interdisciplinary oral healthcare professional. I am an academician by passion and a general dentist and a geneticist by profession. Well, at Indian Academy for Clinical and Dental Genetics, I believe in the motto to create, collaborate, and contribute to science. With a small prayer to my father, Dr. Kamalesh Chandra Roy, who was the first dentist of district Murshidabad in West Bengal in India. Today, let me start the session. First of all, I thank to all of you for being here, friends who have joined from different parts of the world at different time zones. Thank you so much for attending this webinar. Well, let me just introduce Saliva Annex to you. Saliva Annex is a monthly free webinar series, where we will try to cover everything on the fascinating field of saliva diagnostics. This webinar will try to focus on all aspects of saliva diagnostics and its applications in various fields. Through Saliva Annex, we'll also try to throw light upon career prospects, newer career prospects, which you can take up or you can run it parallel with whatever you are doing right now. Research opportunities as well as entrepreneurship opportunities with saliva diagnostics. Okay, so let's get into and get introduced to saliva and saliva diagnostics. So today is the first webinar of the monthly series of Saliva Annex. As rightly and very rightly said by Dr. Irwin Mandel, saliva lacks the drama of blood, the sincerity of sweat, and the emotional appeal of tears. How wonderful it is. How wonderfully described. So, the question is, what is saliva? Well, saliva is a natural fluid that moistens our mouth, supports our ability to eat and speak, and can keep our mouth and ultimately overall body healthy. According to British Medical Association, if we want to define saliva, it is a watery, slightly alkaline fluid secreted into our mouth by particular glands which are present in the human system, in the mouth, that is salivary glands, and also by the mucous membrane that lines the mouth. Why is saliva so important? Why are we trying to talk so much about saliva today? Well, if you see that our spit or saliva contains n number of biochemicals, which may be proteins, DNA, RNA, or anything, meaning to say it contains amounts of biochemicals which are found in the blood and get filtered into the mouth. These proteins and biochemical substances can be linked to different health conditions, not only pathological, it can also give an impression of whether the body is at health or not. Over a period of time, if we can study of the increased or decreased concentration of these elements, these can make saliva a promising diagnostic fluid that has multiple advantages over blood. What is saliva diagnostics? Well, to put it very, very simply, saliva diagnostics is basically the study of your spit or saliva. what scientists do is try to look for certain elements and compounds that can help them diagnose and predict certain health conditions. Till now, you might have been familiar with the word biomarker and you might have heard a lot about them. So what are biomarkers and why are they needed after all? Biomarkers are nothing but traceable substances that exist in saliva. And these substances can help detect and even predict certain diseases. So, for any biological fluid to become a diagnostic medium, biomarkers are needed. So these are measurable, reproducible biological parameters used to gather information about health conditions, both normal or physiological and pathological. So who can be or what can be biomarkers? Biomarkers can be any kind of molecule. It can be a protein, RNA, any metabolite, DNA, any microbial flora that is present. And basically, any molecule which has the potential to serve as a biomarker for any state of disease. In that way, if you see that carriers for these biomarkers can be every human tissue of fluid. The point is not about finding out the presence or absence of a particular biomarker in saliva, but the greatest challenge is to detect and identify the most discriminatory biomarkers in saliva with which we can specifically diagnose and predict about a particular condition or the normal physiological condition. Okay, let's see the war versus saliva and blood. So definitely there are advantages of saliva over blood. With common sense, we know that, if you ask blood from a person, a normal person or your patient, you know the reaction well. So in that way, saliva collection is undemanding. If you ask for 2 ml or 5 ml or even 20 ml of speed from a person or your patient, they even don't bother to understand why you are asking. The procedure is non-invasive. Very, very important, friends. It's a no-needle procedure, which makes sample collection very, very easy. Third, there is no need for any trained medical staff to collect saliva sample. It can be done by anyone. We don't require any phlebotomies that we require while drawing blood. Also, another advantage is multiple samples can be very easily obtained. You took saliva sample, you did some mistake, you throw it up, you ask the patient to give the saliva again, he will never get even irritated or bother to understand why you are doing so. But the condition, you know how it goes with blood as a sample. Some saliva samples are safer to handle. It's very easy to ship and store them. And the very important point is there is minimal risks of cross-contamination. Okay, so we are talking about saliva, so we definitely need to understand from where this saliva is generated inside our mouth. Basically, saliva is generated by special glands that are present called as salivary glands. And in those glands, there are as seen as cells, particular cells, and small tube-like structures, like see this kind of ducts. These are tube-like structures. So all these are saliva is generated by these glands, salivary glands, and subsequently through the ducts, it gets inside our mouth. We have 3 major salivary glands: the parotid gland, the submandibular gland, and the sublingual gland. All these 3 major salivary glands contribute 90% of total saliva, whereas there are other minor salivary glands like labial, buccal, lingual, palatal, etc., which supplies the remainder. If you see the composition of saliva, it's so fascinating. The 99% of saliva is just water. However, you put 99 or 99.5% is water. Only 1% or 0.5% is composed of solids, and you just see what these solids can be. This can be organic substances, inorganic substances, gases. Just glancing at this chart seems that saliva can have everything what that can be present on Earth. How does it happen? As I mentioned earlier, that saliva is a filtrate of blood. So there, through these common mechanisms of diffusion, active transport, ultrafiltration, transudation, biomolecules keep exchanging from blood to saliva. This is how it makes saliva such a rich biological fluid. Now, if we try to understand different types of saliva, we can broadly classify as gland-specific or whole saliva. Whole saliva is nothing but a mixture of all the various glandular salivas produced in the mouth, along with varying amounts of other components. Like as you can see in this picture, it can have some microbiomes, it can have other fluids like nasal secretions, it can have certain blood derivatives. Well, the composition of whole saliva is not constant, and it changes depending on the degree of stimulation present in the various glands. What do we mean by gland-specific saliva? As I told you about the major salivary glands, so each salivary glands have different types of combinations or ratio of certain cells called as serous and mucous cells. So these different ratio of serous and mucous cells are basically important, and they make the different types of saliva, and we classify saliva as serous, viscous, and mixed. Like for example, the secretion, the salivary secretion from parotid gland would be kind of serous, that means more watery. Sublingual secretion would be viscous because it has more amount of mucous cells and it produces a compound called mucin. So this will make the sublingual saliva kind of viscous. The secretion from submandibular gland is mixed. It has both serous and mucous cells or mucous acini cells in kind of equal proportion. Now I will tell you about very, very important aspect or the considerations regarding the collection and sample processing steps of saliva. First is the subject status. It has been seen, it is very important to consider whether the person is fasting or he has had a full diet, because it has been shown that the content of saliva, the quality or composition of saliva differs with the person is fasting. Collection time, though there is no gold standard rule right now, but sample collection time, it has been seen that saliva collected in the day, midday, or night differs slightly. So we need to actually form or work on the process where we can come up with gold standards of when to collect saliva. The volume requirement, depending on what kind of study you are going to do, depending on what kind of pathology you are dealing with, sample collection volume is another important aspect to be considered. Method of sample collection again is very, very important depending on the downstream processing, and not only that, what kind of case or patient you are dealing with. And last but not the least is sample processing and storage. There are now, by research, commercially available n number of salivary diagnostic tools, like the picture that you can see here, for different purposes. Like, for example, PureCell, it is a device for specific collection of RNA or proteins from the saliva. Then you have something called as microsal, which can be collected, used for saliva collection from small animals, and PDSL for collection of saliva from infants, so on and so forth. So just imagine, I mean, that's so wonderful. Nowadays, we have so many things available. Well, there is a common confusion to understand whether saliva diagnostics is a genetic test. Yes, some tests are, but not all. The type of testing where scientists analyze the DNA or genetic descriptors a person may have in their saliva to either confirm or predict a disease is considered a genetic test. Oftentimes, the testing of saliva is used as a diagnostic test to confirm a diagnosis when a person has signs or symptoms of a possible disease. Yet, the testing of saliva can also be considered a predictive test that can show which individuals have higher chance of getting a disease even before symptoms appear. And that's the wonder of genetic testing. Can children take up this test? Yes, anybody can. Young or old can be tested. Since it takes just a simple cheek swab or droplet of saliva and it's non-invasive, it's painless, so there's no reason to fear taking saliva samples or taking up any tests pertaining to saliva. Okay, so let me just tell you where have we are right now at present, from lab to bench to chair side, the journey. The major technical roadblock for saliva diagnostics at this point of time is that all saliva samples have to be kept on ice, because we certainly do not process the saliva samples as soon as we collect most of the time. After addition of RNA and protein stabilizers, most of the time the samples has to be kept in minus 80 degrees centigrade until analysis. So this procedure of storage is definitely well suited for research purposes, but obviously not for saliva testing in dental offices or low to medium equipped medical facilities. So for that reason, customizing saliva sample to be more convenient and less sensitive to ambient conditions is very, very necessary. So let's see what we have in hand right now. Presently, we have different point of care saliva diagnostic platforms. For example, we have an oral fluid nanosensor test device, or OFNASET, which has already got FDA approved and should be available soon. It is a handheld device and you can carry it anywhere. Another fascinating device is Lightbox Image Analysis Module, or LIAM, with which again you can take this device anywhere and you can analyze saliva cortisol, stress biomarkers and others, and immediately you get the results on your smartphone by connecting both the tools on the devices, your smartphone and the device through Bluetooth. Wonderful, isn't it? I will tell you an interesting fact that, so how far have we come with saliva? Now, at present, we have something called as Saliva Wix Knowledge Base, or SKB. It is a web-based data management system dedicated to help clinicians use saliva as a diagnostic tool. It has been developed by Dr. David Wang's lab of the UCLA School of Dentistry. And now with SKB, basically you can explore the biology, diagnostic potentials, the pharmacogenomics of human saliva. This system allows approach to the utilization of saliva diagnostic technology for personalized medical applications. Well, from our side, we did a small effort to conduct Saliva Symposium India, the first international summit calling in all saliva experts right here in India in January 31st this year. I welcome all of you to be a part of SALSI, or Saliva Symposium India 2020, which will be coming soon, and we will be doing it in Bangalore this time. Another workshop called Saliva Annex 2019, which is going to happen in July 14th. It is a one-day workshop on saliva diagnostics and genomics, where we will be telling you from the very basics of genetics to wonderful genomic tools, how you can use them in your daily diagnosis or daily research. And there will be live demonstration of the Liam device that I just showed you. This is going to happen in Times of India, Bangalore, on July 14th. In the description box, all the links are given to register for this particular event. Also, if you are interested to see about Saliva Salsi, please click those links and whenever you're free. Well, to summarize, the evolution of saliva diagnostics has reached a new level toward the goal of using saliva as a powerful fluid from early detection and the first line of diagnosis for life-threatening diseases such as cancer, we have metabolic disorders, any kind of infections and inflammatory diseases. Newly developed tools, the ones that I shared with you, like the point of care devices and about the SKB, the database of saliva diagnostics, are helping to realize the goal of making salivary diagnostics available to clinicians worldwide. This is a unique moment, and I am not biased, my friends, where dentistry may be paving a new path for primary health care. So, what can we expect in the future? Building on the research of today, saliva has the potential of becoming an even more commonly used diagnostic fluid. Nowadays it can detect gum disease, multiple cancers, breast cancers, for example, oral cancers, even heart diseases, diabetes, and whatnot, and many more health conditions that can substantially impact our life. So it's very, very important that we join hands together. We become interdisciplinary and we take part in the journey of saliva diagnostics to make it more popular, to make it a habit to practice saliva diagnostics. I thank my mom for bringing me to this world, my husband, the Braj, and my lovely girls, Shruti, Namrata, Roshni, and Kanchan, who always help me in all my endeavors. And thank you so much to be in this webinar, and I look forward to see you all in the future webinars. We will be meeting every month from